primarily to its structure, characteristic features, and cellular dynamics. Sir explained an interesting fact that Hydra is potentially immortal organism which escapes again and refuses to die through mechanisms of its stem cells and its regenerating capacity by illustrating few experiments on Hydra. Respected Dr. Susan Evan, Scientific Officer retired H Integrate Head Plant Biotechnology and Secondary Production, Baba Atomic Research Center, Mumbai, has clearly explained the journey of biotechnology by briefing out various milestones in biotechnology, starting from Mendel's experiment, DNA, fingerprints, penicillin discovery, PCR, gene therapy, plant tissue culture, to human genomic project. Madam explained on crop improvement for feeding the world by explaining the importance of green revolution, which made poor country from begging to supplying nation for wheat. Madam even stressed on smart agriculture and advantages of it on the future agriculture. Respected Dr. Rita Mulhekar, Fellow of National Academy Sciences, retired scientific officer, H in grade, ATREC, Tata Memorial Center, Karagar, Navi Mumbai, delivered an elaborating lecture on genomic and molecular medicine. Madam delivered and explained goals and promises of a human genomic project. She even stressed on microarrays and next generation sequencing techniques, pharmacogenomics, and genomic medicine. Respected Dr. Ahmad Abdul Halim Khan, sir, Chairman, Board of Studies, Department of Botany, Telangana University, Nizamabad, has explained in detail about developments in science and technology in India after independence in different decades, starting from eradication of smallpox to green revolution, vaccine manufacturing, and origin of prestigious research institutions and pharma companies in India, and made us to recollect the contributions made by eminent scientists from our country. I sincerely hope that the ideas and views during the interactions will sow its seeds for sustained, continuous, and many more meaningful workshops. I welcome you all to share your knowledge and vast experiences with the learning community. I wish everyone a successful and fruitful lecture workshop. I thank you all for your kind participation. Thank you.
Thank you, ma'am, for the warm embrace. With no further delay, let us begin the session. I request our speaker, Honorable Dr. Surendra Kaskadbi, to continue the day 2 session. Now I request Sir to take over the session.
Okay, good.
Thank you, sir.
So I'll share my screen first
Okay, sir.
and just confirm if you can see.
Okay.
Can you see the first slide?
Yes, sir. Yes, sir.
Okay. Okay, so good morning and welcome to the second day of this lecture workshop organized by the college, which has been organized very well and funded by the Academy's education program. I was just talking to Dr. Rita Mullerkar and she told me that all the lectures went off very well yesterday and the students were very participative. They asked good questions. I apologize for not being there all the time because I had some other things lined up. But Dr. Rita assured me that it went off very well. So today we'll start a session again with the same series of speakers. I'll speak first, then Dr. Susan Apen, then Dr. Rita Mullerkar, and finally Dr. Halim Khan. Today what I'm going to discuss is about how form and shape develops in animals. So this is just a picture from the internet wherein it is seen that there are different kinds of organisms that you see in nature. And if you look at, I have picked up some interesting pictures from the internet, and you can see here the various kinds of animals that we come across. These are very spectacular forms and shapes of animals. And so you have snakes, you have birds, you have mammals, and dolphins, and so on. And we come across all this animal diversity all the time. What you see at the right hand bottom corner are two specimens of the species Homo sapiens, our species. Because of my age, I have taken Sachin Tendulkar and Amitabh Bachchan's picture, but you can take somebody who's younger, maybe from Bollywood and from the cricket fraternity. What we see from these two picture of these two gentlemen, Sachin Tendulkar and Amitabh Bachchan, is that in spite of having the same human genome, they are so different. Their heights are different, their achievements are different, their looks are different, and various kinds. So two individuals, all of you sitting in the room, unless there is a case of identical twin, in spite of having the human genome, we are all different. However, today I'm not going to consider why we become different, but what I'm going to try and convey to you is how these different kinds of animals evolve, in spite of having the same kind of genetic toolkit that I was discussing yesterday when I talked to you about Hydra. Okay, so uh, all these animals we come across, but it is impossible to study all of them. It's not possible to, for example, all the time work with tiger development, because you will end up in his stomach rather than studying it. So what scientists usually do is that they choose certain organisms which are good to be brought to the lab, and they are called as model systems. So these are research organisms which offer certain advantages in our study of biology. What I have shown here on the left of the screen are 6 very popular model systems. A is a worm, a tiny worm which grows in a petri plate, feeds on bacteria. This is known as Caenorhabditis elegans. Now this entire worm, the adult worm, is made up of only about 1,000 cells. Please remember, when I was talking about Hydra yesterday, I said that a Hydra is made up of 100,000 cells, and we are made up of trillions of cells. But this tiny worm is made up of just about 1,000 cells, and therefore it is easy to study how all these 1,000 cells develop from a 2-cell embryo, which is shown in the first part of the panel. Okay, so it is possible to label individual cells in the early embryo and find out which are their progeny in the adult life. So, for example, if you label the cell on the left by some means, and if the label is fixed in that cell, all the progeny of this cell. So this cell will divide into 2, the 2 will divide into 4, 8, and so on. And all the cells which come from this cell in the adult worm will have the label. So you know that which cell has arisen from which original embryonic cell. This is known as cell lineage studies. For example, in our body, we would like to know where the skin cells arise, where are the stem cells, and so on. So these kind of studies are very useful in case of worm. The other model I have shown here in panel B is the famous model Drosophila fruit fly. As you know, fruit fly Drosophila melanogaster. And this has been the most powerful model system in genetics and development. The advantage with Drosophila is that you can easily bring them to the lab, you can maintain them, and they have a fast generation time. So if you induce mutations in individual genes, you know what is a mutation? By inducing mutation, you change the structure of the gene so that the function of the gene is affected. By looking at the uh, adult organism coming out of the mutant embryo, then you know. For example, if you make a mutation in a particular gene, and if the fly does not develop a wing, then you know that the gene is important for wing formation. So likewise, you can induce mutations one gene at a time and then find out what is the function of the gene. This has been very popular model system because it has been used for more than 100 years and is very widely used all over the world, mainly because you can induce mutations. The other two model systems which I have shown here in panel C and panel E, okay, these have been traditionally very popular. Excuse me for a second. Sorry about that. So these have been traditionally very popular. The one in C is the frog embryo, and the one in E is the chicken embryo. So frog embryos are readily available in ponds, at least they were. And then our knowledge about gastrulation, organogenesis, that means organ formation and so on, has come by studying the frog embryo. On the other hand, the chick embryo has also provided us with similar information, but chicken embryos were always available to the scientists because poultries were there. So chicken embryo, for example, has been used even by Aristotle. So old naturalists, they were not really called scientists, also used chicken embryo for their work. So because frog and chick are the vertebrates, they are more close to us, and we know more about our own development by studying vertebrates rather than studying invertebrates like the worm and the fly. Another model system, vertebrate model system, which has become very popular in recent years, is the model system shown in panel D. This is zebrafish. So zebrafish looks like a zebra. It has stripes like a zebra. It's basically an Indian fish found in the Ganges. And what you can do with this is that you can induce a lot of mutations. So inducing mutations in frog and chicken embryos is relatively difficult. And therefore, if you can induce mutations in a vertebrate embryo like the zebrafish embryo, just like Drosophila, you are able to learn a lot more about normal development. In addition, the embryos are quite transparent and you can study the development of various structures under the microscope. Relatively, frog and chicken embryos are non-transparent. And finally, in panel F, you see a mouse embryo, because ultimately we are mammals and therefore working with mouse and rat embryos will tell us more about our own development. These embryos are of course difficult to work with because as soon as you take them out of the body and grow them in vitro in the lab, uh, things are different because usually they grow inside their mother. But it depends on what kind of question you are asking. It depends on what kind of inquiry you are making. You choose a model system which is more suitable. In addition to these 6 model systems which are very popular, there are 3 very simple model systems. So sponges, which are shown here, red sponge, then another organism called Dictyostelium, and of course I have told you about Hydra. So just briefly, sponges belong to phylum Porifera. They are poorly organized, and you can study cell disaggregation and cell aggregation by using sponges. Dictyostelium is an organism which shows alternation between unicellularity and multicellularity. So when the food is available in plenty, they survive as unicellular amoeba. But when food becomes scarce, the amoebae come together and form a multicellular organism. And this organism can move from one place to another. And you can see it is moving. So thousands of amoebae come together, form a multicellular body, and then it can actually differentiate. That means it can form spore cells on the top and stalk cells at the bottom. So if you remember our yesterday's discussion, or when you look at embryonic development, you see that there are many of these processes happen in embryo. So let me briefly tell you what I mean by that. So all of us begin our development as a unicellular zygote. Okay, so please imagine your own development. You started your life, on this earth when the oocyte from your mother was fertilized by a sperm from the father, and it became a unicellular zygote. So all of us start as a unicellular zygote, which is as big as a full stop in your book. Okay, so we are so tiny when we begin our life. This zygote undergoes many cleavage divisions, and they become like a ball of cells. It is like a strawberry, or you must have seen a bundi ka laddu. So we all look like a bundi ka laddu when we are small. Okay, and then this ball of cells has to be converted into an organism. For example, imagine me as a small ball of cells, and then you have to make an anterior and a posterior. You have to make a dorsal, a ventral. You have to make a left and right, and so on. So to make this ball of embryo has to be converted into a 3-dimensional organism. So what we are going to do now is to see how this is actually done and how this is regulated at the level of genes and at the level of cells. So I'm going to take examples of 2 Nobel Prizes in medicine to explain this phenomenon. Sorry. Okay. So the first Nobel Prize that I'm going to discuss is a Nobel Prize in Medicine in 1995, which was given to these 3 individuals: Edward Lewis, Christian Nelson Vollard, the lady, and Eric Wishaus. So what they found out was that they discovered the genetic control of early embryonic development. So I was mentioning to you about Drosophila. These people find out how a Drosophila development happens and how it is controlled at the level of the genes. So let me explain the model system in a little more detail. I just want to ask you whether you can hear me and whether you can see the slides.
Yes, sir.
Okay. Okay, thank you. So this is a Drosophila fly. It's a simple fly which you often find in your houses on a rotten banana. If you leave a banana to rot, then you will see a lot of flies. So what you need to know to begin with is that this Drosophila has three main parts of the body. One is the head, the middle is the thorax, and then is the abdomen. So it is a three-part body that the Drosophila has. Then it has the dorsal axis on the top. So our dorsal is here at the back, and it has a ventral axis at the bottom. So our ventral is in the front. This is the anterior. So this is the head. Our anterior is here, and posterior is in the foot. So this is dorsal, this is ventral, this is anterior, this is posterior, and it has head, thorax, and abdomen. Now you will also notice that the body of Drosophila is segmented. You can clearly see the segments in the abdomen, but the entire body is segmented. So our body is also segmented. I'm not talking about six packs. I'm talking about, if you look at the ribs, if you look at the vertebra, you will immediately know that our body is also segmented. So the entire body of Drosophila is made up of 14 segments, 1-4. Okay. There are 3 segments of the head, which are known as head segments, 3 segments of the thorax, which are known as thoracic segments, and 8 segments of the abdomen. So there are 14 segments of the body. Now the question is, how different structures develop at particular places on the body. How my hand has developed here and not somewhere else. So, for example, if you take the wings of Drosophila, Drosophila has only one pair of wings, unlike butterflies, which are also insects. So, its wings are developed in the second thoracic segment. You can see here, they are coming out of the second thoracic segment. This is the wing on our side, this is the wing on the other side. You can notice a small bulbous structure here, which is known as haltere. It is a balancing organ. Now, please remember that the wings develop in the second thoracic segment and the uh, halteres develop in the third thoracic segment. So, for example, you can ask a question as to why the wings do not develop either in the first or third thoracic segments, or why don't they develop on the head or on the abdomen. So these kinds of questions, the 3 scientists who got Nobel Prize tried to answer. You will also notice that being an insect, Drosophila has 6 legs. All insects have 6 legs. That's why they are called as hexapoda. So you will see that every thoracic segment develops one pair of legs, and that's how Drosophila has 3 pairs of legs. Then the Drosophila head has eyes, antenna, and so on, the mouth parts by which it feeds. The abdomen has other structures in the body. So this is the basic structure of Drosophila. Now I'll quickly go through how the early development takes place. Okay. So in case of Drosophila, this is the fertilized zygote. And in the early development of Drosophila, the nucleus goes on dividing. As you can see, from 1 to 9, the nuclei go on dividing. However, the cytoplasm does not divide. So it's a syncytial embryo or syncytial blastodon. I hope you get this. See, the nucleus is dividing, but the cytoplasm is only one. So all the nuclei are present in a single cytoplasm. So it is known as a syncytial blastodon. But at some stage of development, at stage 10, for example, these nuclei move towards the extremities, move towards the side, towards the border of the embryo, and then a cell membrane develops between adjoining nuclei. So one cell will have one nucleus, just like we have. But in Drosophila, initially, it is syncytial, and then it becomes one nucleus in every cell, in which case it is now known as cellular blastodon. So syncytial blastodon becomes cellular blastodon. Why it is important for Drosophila is because if you look at this embryo, for example, in 8, 9, or even in 7, if you have some molecule at the top, some chemical at the top, you can imagine that it can freely diffuse from this to this end because it is only single cytoplasm. If it has to happen in a cellular blastodon, every time the molecule or the chemical has to pass, it has to cross membranes of the cells. That is not necessary for the syncytial blastodon. So the earlier signaling mechanism in Drosophila happens because of diffusion of chemicals, which is much more simple. I'm going to summarize these findings, and this is a summary slide before I go to the details. So what these 3 Nobel laureates found out is that there are certain groups of genes which regulate Drosophila development. So one is a maternal effect genes. That means the mother herself deposits certain mRNAs, and then it leads to formation of protein gradients, which leads to gap genes. Gap genes means if these genes are mutated, there are gaps in embryos. Then pair rule genes. That means you can see, I told you that Drosophila has 14 segments. So you see 7 stripes here. So these decide pairs of segments in the body. And finally, there are segment polarity genes. So I told you that Drosophila has anterior, posterior. So every segment also has an anterior polarity and a posterior polarity. So certain genes decide that. And finally, you have a group of genes which are known as homeotic genes, which decide which structure will develop on which segment. For example, it will homeotic gene will decide that the wings will develop only on the second thoracic segment and not on any other segment. So these groups of genes were discovered by the 3 Nobel laureates. Let me now take some examples. So this is the early development of Drosophila. The yellow cell here is the oocyte. So you can see that the oocyte is developing in size. It is growing in size, and finally it becomes big. What I would like you to notice before, because going into details is not necessary. What I would like to notice at in the bottom figure here is that you see certain red molecules deposited on one side of the oocyte and blue molecules deposited on the other side of the oocyte. So this is an oocyte, and these molecules are deposited by the mother herself. The fertilization has not yet taken place. So when the oocyte is formed, it has polarity, in the sense it has unequal distribution of chemicals. On the left hand side, there is more red molecule. On the right hand side, there is more blue molecule. So what happens with the red molecules is that this is the mRNA, messenger RNA, which will get translated into a protein. So messenger RNA is deposited on the left-hand side of the oocyte. This gets translated into a protein through the process of translation. And you can see that the protein is formed. And because the cytoplasm is one, if you remember, I explained to you that these are syncytial blastoderms. So because the cytoplasm is one, it can easily diffuse from high concentration to low concentration. These are through simple laws of diffusion. So as it diffuses from high concentration to low concentration, it forms a gradient. It forms a concentration gradient. So if you look at the color here, you will see that it's highest in concentration on the left-hand side, and it goes on reducing in concentration as you go towards the right. Okay. And it so happens that wherever you have the protein, red protein, but its name is bicoid, by the way, you need not remember it, you can read it up later. So wherever bicoid is present in large quantity, that will become the head of the Drosophila. Okay. So the anterior part of the Drosophila is decided by the bicoid gene. Now, if you mutate this gene, bicoid gene is mutated, that means it cannot function properly. It cannot function properly in the sense that it cannot form the bicoid protein. Then in that bicoid mutant, you can see here that there is only abdomen. There is no head or thorax. This is the wild type. That means normal fly, which has head, thorax, abdomen. Of course, there are two extremities also, which we'll not consider right now. But in the mutant where bicoid is not present, it forms only abdomen, and the head and the thoracic structures are absent. If you look at the blue embryo, it does exactly the same thing, but in a reverse direction, because blue is deposited on the right. So when the protein is formed, it forms a gradient in an opposite direction. And it so happens that blue is the one which is responsible for formation of the posterior structures. So if you have a mutation in this gene, which is known as nanos, then what you see here is you get a fly which has only head and thorax, but there is no abdomen. Okay. And so now what we have seen is the red protein, which is known as bicoid, is responsible for formation of the anterior structures, head and thorax, while the posterior structures are formed because of the blue gene, which is known as nanos. How do scientists know that the gradients actually form? They have actually seen it by using different techniques. So this is a technique called autoradiography. wherein you can see that the bicoid messenger RNA is deposited only on one side of the embryo. And then this messenger RNA gets translated and the protein is formed. And you can see that the protein gets diffused from left to right and it is expressed in the nuclei. Because bicoid is a transcription factor, you will find it in the nucleus. So you can see that these nuclei on the left hand side have the highest concentration of bicoid. And as you go from left to right, less and less bicoid is present in nuclei, and there is no bicoid in nuclei which are present here. You can actually calculate the amount of bicoid protein in this nuclei and plot a graph. So if you look at a normal fly, which is known as wild type in genetics, then you can see that the left hand side, if you consider it 100% expression, then as you go from left to right, then it goes on falling. But if you calculate the bicoid protein present in the mutant of bicoid, then see the red line. It has very little protein or no protein present, and it is a flat line. It is a flat line which shows that there is no bicoid mutant. There is no bicoid protein. And therefore, in this fly, which is shown in red, head and thorax are not formed. So it is concluded from such experiments that bicoid is the one which is responsible for structures on the anterior side. Now, this is one experiment, but we can do many experiments. And I'm just giving you one example as to how scientists find out what is the precise function of a particular gene. So let's again look at bicoid. So in normal development, bicoid mRNA is deposited on the left-hand side. And then when it is translated into protein and when the gradient is formed, it forms a normal structure: head, thorax, and abdomen. And these acron and telson are the extremities of the body. So this is a wild type phenotype, okay, on the left hand side. Then we considered the mutant where bicoid is not present. You saw the quantity of bicoid protein is almost negligible, in which case you see formation of only abdomen, okay? So you see abdomen only. There is no thorax, there is no no head, and there is no anterior acron also. Only abdomen and two telsons are present. Now, what additional experiments we can do to confirm what is the function of bicoid is that you can take this mutant. So now you have a mutant which is no bicoid, but inject bicoid messenger RNA or bicoid protein from outside. So in a recombinant DNA lab, you can make such things, and using a fine needle, you can inject from outside bicoid into this mutant. And what you see is amazing. As soon as you provide bicoid protein to this embryo from outside, it forms a normal structure. It undergoes normal development. So once again, you have acron, head, thorax, abdomen, and telson. But you can do a little more variation of this experiment. Scientists like to do that to confirm the function of a particular protein or gene. What you can do is, instead of injecting bicoid in the bicoid mutant at the proper site where it is usually expressed, you can inject it in the middle. And now you see the amazing result. If you inject it here, you will get head in the middle, thorax on either side, and little bit of abdomen. So what we are seeing now is, because now bicoid is expressed here, the cells here, instead of forming the middle structures, will go on to form the head in the middle, thorax and abdomen. And finally, an experiment you can do wherein you can take a normal embryo and inject additional bicoid on the other side. Now you get, what you see is one small abdominal tissue in the middle, two thoracic tissues, and two heads on either side. So it is a clear-cut proof that wherever bicoid is in high in quantity, that will form the anterior structures. So these are the kinds of experimental approaches scientists take to find out the function of individual genes during early development. So this is just one example of a gene that they found in Drosophila. Like that, they found thousands of genes which control development in Drosophila, about how anterior posterior is decided, how dorsal ventral is decided, and so on. Now there is another set of genes which is present in Drosophila, and these are known as homeotic genes. So homeotic genes are the ones which decide, as I told you, which structure will develop in which segment. So these are basically 8 genes which are shown here, and they are present in form of 2 clusters. One cluster is known as antennapedia complex. Once again, please don't bother too much about the names. But this cluster has 5 genes, as you can see, and the second complex is known as bithorax complex, a cluster of genes which has 3 genes. What I would like you to only concentrate on is compare the colors. So labial gene is responsible for formation of these head structures, while abdominal A, abdominal B are responsible for formation of such structures. So please remember that the colors that are shown in the adult fly have actually come as an effect of these genes which are shown in the similar color. So what the third Nobel laureate found is that these genes decide which structure will develop on which segment of the body. So you can see 14 segments here. Actually, you can't see the 3 head segment, but 3 thoracic segments have been labeled: T1, T2, T3, and there are 8 abdominal segments. And you can compare the color code and decide which gene is responsible for development of which structures. How did scientists find it out? They did a number of experiments. So one of the experiment, two or three experiments I'll briefly discuss with you. The one experiment that they did was that this is a normal Drosophila head under a compound microscope, and this is a normal Drosophila head under a scanning electron microscope. So what you can see is 2 red eyes. There are 2 antennae in the middle, and these are the mouth parts. So once again, under scanning EM, you can see the 2 eyes on either side. There are 2 small antennae here, and below are the mouth parts. Now, what one can do is one can express different genes at different places of the body, just like the experiment that I showed you about bicoid. So what in this experiment, what these people did is they expressed a gene which is responsible for leg formation on the head of the Drosophila. Please understand the expression. Legs are usually formed in the thorax, but these scientists expressed leg forming gene in the head of the Drosophila, and you can see that this Drosophila has developed legs on its head. Can you see these legs very clearly? So instead of antenna, these Drosophila have developed legs. Now this is an experiment which is done in the lab. You will not usually find such mutants in nature. So don't go out looking for flies which are walking on their head. That doesn't happen. But what these experiments tell us is that the function of the gene, so this gene which is expressed in the head and which gives rise to legs in place of antenna, we know that it is important in leg formation. Another example. So this is a normal Drosophila. I told you that the second thoracic segment of Drosophila has 1 pair of wings. You can see the left wing and the right wing. And the third thoracic segment has a balancing organ called halter. So these are the 2. And so what people did was they expressed the wing forming gene in the third thoracic segment so that they got a fly with 4 wings. So now the halter in this case converted into a wing because the gene is expressed there. On the other hand, they also formed a fly which has 4 balancing organs but no wings. So this is a fly which has 4 wings. It can fly, but it cannot balance itself because the balancing organs have been lost. This fly cannot fly because it does not have wings. But what these experiments tell us is that which gene is responsible for formation of which structure. They have also produced an eye on the leg of the Drosophila. Can you see this? This is a leg of the Drosophila. If you express a gene responsible for leg formation, you can get a eye on the leg of the Drosophila. And some scientist was not satisfied with one eye, so he actually made many eyes on the back of the Drosophila. So there was a Drosophila fly with hundreds of eyes on its back. So you can see these red eyes. Of course, the eye here or the eyes here cannot see because there is no innervation there. They don't go to ganglion. These are only structural components. So they look like eyes, but they cannot see. So all these experiments tell us which gene gives rise to which structure in the particular segment of the body. Why would this work get a Nobel Prize in medicine? Because this is work in Drosophila. This is a small, tiny fly which is found on rotten banana. Then why these people should get a Nobel Prize in medicine? Because medicine is usually considered to be only for humans in this case, in the case of Nobel Prize. So what later on scientists found is that these are those 8 genes that I showed in Drosophila earlier. 1, 2, 3, 4, 5, 6, 7, 8. So 2 complexes. What people found out later on by using bioinformatics and computation analysis is that mammals also have similar genes. So mouse also has similar genes, but we also have similar genes. But what has happened during evolution is that mammals have four sets of such genes. So Drosophila has only one set of such genes, but we have four. So we have, these are known as Hox genes. So there is a Hox A cluster, Hox B, Hox C, and Hox D. So we have four sets of those genes. This has probably happened during evolution because of duplication of the chromosome twice. Uh, and what is the advantage? Because we already know that if A1 becomes mutant and it cannot carry out its function, then B1 can take over its function. So it is a kind of insurance against abnormal development that we have probably developed four sets of these genes, which are homeotic genes. And obviously, when you see abnormalities in human beings, then if you can study the genes which are responsible for the abnormalities, you can treat the disease or you can avoid, such abnormalities. And therefore, this work ultimately got a Nobel Prize in medicine. This is a comparison between similar genes in Drosophila on the top and vertebrates. So just compare the color codes and see that we are not all that different from Drosophila. Yesterday, I tried to convince you that even Hydra has genes similar to us. So we are not very different from Hydra either. So what I'm trying to convey to you is that the so-called genetic toolkit evolved only once, and it has been used again and again for different uh, formation of biodiversity. Now, let me jump on to the other Nobel Prize. Okay. So, this is another Nobel Prize in medicine. I have I have completed with the first Nobel Prize, wherein we saw how scientists discovered genes which regulate not only Drosophila development but also human development, and that got them a Nobel Prize in medicine in 1995. Now, this other story that I will complete in the next 15-20 minutes is about a Nobel Prize in medicine that was awarded in 2012. It was awarded to two gentlemen. One is Professor John Gurdon, who is shown on the left, and the other is Professor Shinya Yamanaka. Okay, and I will briefly describe their work to you. So what they did was they discovered that mature specialized cells can be I will explain this, can be reprogrammed to become immature cells capable of developing into all the tissues of the body. Their findings have revolutionized our understanding of how cells and organisms develop. So let me develop this and explain what is meant by this Nobel Prize citation. So I told you that we are initially like a ball of cells, and then we become a three-dimensional organism. But initially, our cells are also similar to each other. They are not different. They become different as we develop. So some cells will become neurons, some cells will become skin cells, some cells will become cardiomyocytes in the heart, which help the heart into beating. Some cells will become pancreas. Even amongst pancreas, there are some islets of Langerhans. There are other cells which secrete the enzyme amylase. There will be liver cells and so on. So we have different kinds of cells. And as development progresses, our cells start becoming more and more specialized. If you remember, yesterday I explained to you the concept of stem cells. So initially, cells in the early embryo are like stem cells. They can form many different kinds of cells. But as you go from early development, let's say blastula, gastrula, this is in case of frog, you see that as you go from earlier development to adult, you will see more and more cell types are formed. And this happens because the cells become more and more specialized. But and you must remember that even today in my body, all the cells have the entire genome. So all the genes are present, but many of the genes are switched off. They are shut off because they are not necessary for those cells. So this process of cells becoming specialized is known as cell differentiation. Okay, we touched upon it yesterday, but I'll explain it to you again today. So as development progresses, cells start becoming more and more specialized, a process called cell differentiation. Okay, and this cell differentiation is achieved because of differential gene expression. What do I mean by differential gene expression? I just told you that every cell in our body has all the genes. But all the genes are not required in all the cells, because ultimately genes code for proteins. So, for example, the cells in my body which are not in islet of Langerhans do not need to express the insulin gene, because they will not make insulin. Insulin is made only in a subset of cells in pancreas. So the insulin gene in my skin or in my brain or in my hands is switched off because it is not functional. So when cells become more and more specialized, they switch off genes which are not required. And that is why you see that if you take a liver cell and if you take a pancreatic cell, they express different genes, which is known as differential gene expression. And why differential gene expression is because the cells require different genes, different proteins. So if you take a single cell in the body, it will require two kinds of proteins. One is housekeeping proteins. What do you mean by housekeeping? They are necessary for the survival of the cell. Like, cells for genes, proteins for respiration, proteins for metabolism, they will present in all cells in my body. Okay, because the cells have to be alive. But specialized genes will be expressed in only certain cells. We took the example of insulin. So insulin gene will be expressed only in the islets of Langerhans, and insulin protein will be made only in the islets of Langerhans cells, not anywhere else. So likewise, cells become differentiated. Now let's go to what Professor John Gurdon did. His experiment I will explain. This experiment he did sometime in the 1950s. So we are talking about an experiment done over 70 years ago. What he did was he took two frogs, okay, frog A and frog B. From frog A, he took the egg, okay, and from the egg, he removed the nucleus by using radiation. So he destroyed the nucleus and he got only the egg which has no nucleus. Then he took frog B. From frog B, he took a differentiated skin cell. So he took a skin cell, he took out the nucleus from the skin cell and put it in the egg of this frog from which the original nucleus has been removed. And what he found to his amazement, that he can actually get a clone of frog B. So he got a clone which was identical to frog B. And why this was like that is because the nucleus came from frog B, and therefore he got a clone from frog B. So these were the kinds of experiments that John Gurdon started doing. Now, to confirm it further that the nucleus is responsible, and the nucleus, the clone is of the frog which donates the nucleus, he used a wild type frog and an albino frog. So there are albino frogs available. So what he did was he took a nucleus from the albino frog, okay, and put it in the oocyte of a wild type frog after reviewing its own nucleus. And he got so many clones of the albinos. This was a very major discovery in 1960, 1959, 1960. And it was so different that nobody would accept that it is true. Because that time, nobody thought that once an animal develops, its nucleus can actually give rise to a clone. So what John Gurdon did was, he kept his clones in Cambridge, United Kingdom. He used to work in Cambridge, United Kingdom, and went for some work, postdoctoral fellowship to United States of America for 2 years. Okay, in that 2 years, he didn't do anything about this work. After 2 years, when he came back to Cambridge, he found that all his clones were not only alive, but they were reproducing. So these were true clones. And then in 1962, he published his work. That is the first case of animal cloning. So Dolly came much later, but John Gurdon is the first person who cloned the entire animal, and published it in 1962. Please remember that John Gurdon had to wait for 50 years to get Nobel Prize for his work. So his work that was published in 1962 got him a Nobel Prize in 2012. And the same technique was used for formation of Dolly. And let me quickly explain how Dolly was cloned. So this is a sheep, which is known as ewe. So this is a one kind of sheep, fin dorset donor sheep, which gave the nucleus. Okay, right? They isolated a mammary cell from here and took the nucleus. There is the other kind of sheep, old dorset sheep, which donated the enucleated egg. They fused the two, and these fused cells were then gave rise to embryo with multiplying cells. This multiplying embryo was, transplanted into a surrogate mother. That means the mother who is going to allow the embryo to grow. So please please notice here that this is yet another kind of sheep. Why they are using three different sheep? They don't want to confuse with the genetic material. So the genetic material has been donated by the sheep which has donated the nucleus. The cytoplasm came from here, and the embryo actually grew inside the womb of Scottish black-faced ewe. And finally, you get a clone. Dolly was cloned, and which was a clone of the original donor which donated the nucleus. So essentially, they used the technique that John Gurdon perfected in the 1950s and early 60s, and then Professor Ian Wilmette, as you see him here, with Dolly. So Dolly was cloned based on the work that uh, John Gurdon carried out in the last century. Okay, and this technique is used now widely, even commercially. So this is a newspaper report last year, wherein a Chinese shop is actually cloning your pet, cats and dogs, for you by charging you. So you may not be able to see it, but I'll just read it out for you. A dog can be cloned for $53,000 and a cat can be cloned for about $35,000. So actually, by using that same technique, this Chinese firm now clones kittens and dogs. So it is become commercial now. Not only cats and dogs, but a China lab also reported that they cloned lion-tailed macaques, these monkeys they cloned. The one on the left is known as Zong Zong, the one on the right is known as Hua Hua. So they were cloned by using the same technique. So it is probably, in principle, it may be possible to clone human beings also. So that is the importance of John Gurdon's work. Now let me come to the work of Shinya Yamanaka. Okay, I have another 10 to 15 minutes in which I will summarize the work of Shinya Yamanaka and tell you some funny things about Professor John Gurdon. So I told you that our body has specialized cells and which are known as differentiated cells. So once the cell differentiated, they switch off many genes because they are not required. The proteins that come from that genes are not required. So now, if you take a skin cells from my body, then it will express only few genes such as keratin and so on, but it will not express many of the genes. So this is known as a somatic cell, and this is a differentiated cell. What Shinya Yamanaka did was this: he perfected a technique by which he introduced certain genes into a somatic cell and made it a stem cell. I hope you understand what I'm saying. See, stem cells differentiate, then they do not become stem cells. They do not remain stem cells. They cannot divide because they have differentiated. Differentiated cells cannot divide. But what Shinya Yamanaka did is that he used certain genes which are known as pluripotent gene, and he put them in a differentiated somatic cell, and that led to some reprogramming, and this cell, which was earlier a somatic cell, became a pluripotent stem cell. And since it has been induced experimentally, it is known as induced pluripotent stem cell. So you can see the short form is small I and PS capital. So PS capital stands for pluripotent stem and I stands for induced. So now it has become a stem cell. So the characteristics of stem cells I have repeated twice yesterday. They can undergo division, so they can grow in size, but they can also differentiate into different types of cells. So neurons, liver cells, and all mesenchymal cells and erythrocytes and so on. So Shinya Yamanaka found a technique by which he could convert somatic cells into stem cells. So the importance is obvious. See, if you want to, let's take the example of okay, in the next slide I have an example, so let me not jump the gun. Okay, so what Shinya Yamanaka's experiment demonstrated, and this is a very interesting experiment. So you have a mouse here with sickle cell anemia. I'm sure you are aware that sickle cell anemia is brought about by a point mutation. A single amino acid is changed, and after that, the sickle cell anemic mouse has, or human has, cells, RBCs, which look like a sickle, like this, and they cannot carry hemoglobin efficiently, and therefore the individual becomes anemic. So what Yamanaka did was, he took out certain cells from the sickle cell anemic mouse. Let's call him a patient. So he took out the cells and collected skin cells. So skin cells, as I have been telling you again and again, are terminally differentiated cells. So he took the skin cells, brought them to the lab, and put those genes from outside. These are the names of the genes. These are proto-oncogenes. That means if they don't function properly, they can become oncogene and can cause cancer. But they are proto-oncogenes required for normal life. So these 4 genes, pluripotent genes, OCT4, SOX2, KLF4, CMYC. Again, the names you need not remember, these are not important. What is important to note is that if you introduce these 4 genes in the skin cell of this mouse, then you can make the skin cells, convert the skin cells into IPS, induced pluripotent stem cells. Once he got induced pluripotent stem cells, by using recombinant DNA technology, he corrected the mutation. Sickle cell anemia is caused by a point mutation. He corrected the mutation, and after correcting the mutation, he grew the cells in large quantities. So genetically corrected iPS cells were grown, they differentiated into blood stem cells and were injected back into the mouse. And this mouse then recovered for anemia. So this is the importance of Yamanaka's work, wherein you can, for example, there are now experiments going on in human beings, wherein, suppose I am a diabetic, I'm not a diabetic, but let me take my own example. Suppose I'm a diabetic, I cannot produce insulin. Now what people are trying to do, they take skin cells from me, okay, and they convert them into induced pluripotent stem cells, and then again convert them by using different means into islets of Langerhans and transplant them back into me, so that now they can produce insulin and they can cure my diabetes. The advantage here is that since my own cells are taken and they are put again in my body, there is no question of rejection. So immune rejection is avoided when such kind of experiments are there. These experiments are only in their infancy, they are still being tried out, but that is the importance of work of Shinya Yamanaka. Okay, now just to give you a funny side of the story, I told you that John Gurdon published his work in 1962 and he got a Nobel Prize in 2012, and he was born in 1933. He is still very active. He actually works in his lab in Cambridge, UK, Professor John Gurdon, and the institute that he works in is named after him. So it is known as Professor John Gurdon Institute of Developmental Biology. So Professor John Gurdon still works as a scientist there. How old is he? He must be now 1933 to 2021. So you can calculate. Okay. When John Gurdon published his work in 1962, Shinya Yamanaka was born in 1962. Okay. So he's so young. When Gurdon finished his work, Yamanaka was born. Yamanaka did his work and published it in 2006, but got his Nobel Prize very fast in 2012. So sometimes this is a very rare occurrence wherein the Nobel Prize has been awarded so early for somebody's work. Usually it takes a long time. And Professor John Gurdon took 50 years to get a Nobel Prize. Professor John Gurdon's experiments are textbook experiments for developmental biologists. So all of us were praying that he will remain healthy so that he gets a Nobel Prize. You know that Nobel Prize is given only to people who are alive. But John Gurdon is very much active. He comes to India very often. He goes, he likes to go to Himalayas also. So I'll just show you some of his pictures. He has been to India many times. So this is John Gurdon in his own lab in Cambridge. He's playing with the students, just to show you that scientists are also normal. They are not some bearded guys sitting in a closed room and angry with everybody. They are very normal people. John Gurdon is an interesting character. I have met him many times. I'll show you some pictures. So this is an old picture wherein you can see me also and John Gurdon, just to impress you, when he came to Pune in 1993. Okay. Then John Gurdon has been coming to India very often. This was a meeting in Nainital in 1998, wherein you can see Professor John Gurdon sitting here on the right, second from right, sitting in a blue sweater. Okay. Then again, he came to Sikkim Pelling, where you can see John Gurdon sitting right. We are all around here. Many of your teachers will be there. Many of the scientists who are famous in India, many of them are here. This is Professor Obed Siddique, for example, from TIFR. This is Professor Nanjundaya from IISc and so on. So he came also in 2003. He came to my institute in 2008. In fact, he also examined the thesis of a student of mine. But that apart, what I'm trying to tell you is John Gurdon is like a, like a boss in developmental biology, and we were all very happy that he got his Nobel Prize in medicine. I am just going to show you a last slide as a parting gift to you. And this slide is when John Gurdon was in his school, then his teacher wrote, his science teacher wrote a report about him. So when John Gurdon was awarded the Nobel Prize, reporters found that this report, science report of John Gurdon, was on his table. So they asked him why he has kept it there. So he said, that is for my inspiration. Now, I will read the report, then you will know why John Gurdon is so funny, typical British humor, and why did he say that is inspiration. So this is a report of a science report of John Gurdon, which is written in 1949. Please notice that it's a typed report in 1949. The name of the student is Gurdon, division subject biology, and the marks he has got. He got 18 out of 18, 17, 18, 18, 18. But finally, so he stood last in the class here. Here also he stood last in the class, and here stood last but one. So there were 18 students. He got only 231 marks out of 550. So I also got very low marks. So I'm very happy that Professor John Gurdon also got only about 50, a little over 50% marks. Okay. Now read the report. It has been a disastrous half. His work has been far from satisfactory. His teacher is not at all happy with him. His prepared stuff has been badly learnt, and several of his test pieces have been torn over. That means just like we tear our journal papers, he also did that. Okay, so we are all normal. One of such pieces prepared work scored 2 marks out of a possible 50. So once he got 2 marks out of 50, that means 4%. His other work has been equally bad, and several times he has been in trouble because he will not listen. This is very important. He did not listen always to his teacher. People who think originally often do not listen to others. So probably this is very important, but will insist on doing his work in his own way. So he was very adamant about how he will do his work, and that was probably responsible how he went on to get a Nobel Prize, because he thought differently. Nobody thought in 1958 that you can clone an animal, but he could do it because he thought differently. I believe the teacher writes further, I believe he has ideas about becoming a scientist on his presence, showing this is quite ridiculous. So he's actually making fun of the idea that Gurdon wants to become a scientist. If he can't learn simple biological facts, he would have no chance of doing the work of a specialist. And it would be a sheer waste of time, both on his part and those who have to teach him. So this is a terrible report about John Gurdon by his teacher, but ultimately Gurdon kept it as an inspiration and finally got a Nobel Prize in medicine. So, what I would like to tell you is that it may be true that there are books, there are teachers who teach you, people like me who talk to you, but in science, you cannot believe anyone. You have to, see what is the evidence, and you cannot just because somebody is telling you something, you cannot believe. In science, you have to be on your own, you have to be original, and then only you will succeed in science. So I think this is a very good take-home message from Professor John Gurdon. And I think I'll stop here and take questions.
Thank you, sir. Thank you so much. Now I request the participants, those who have queries can post here. Participants, those who have queries can unmute yourself and ask the questions. The session is open for queries now.
ಮತ್ತೆ ಮೂರ್ತ ಶಬ್ದ
I've stopped screen sharing. Is there any question?
Sir, thank you so much, sir. If you participants are requested to post the questions in the chat box if you have any. And really, it's an interesting lecture, sir. So you have delivered a valuable information related to developmental forms and shapes in animals.
My pleasure. My
Sir,
pleasure.
you have explained the genetic modifications in Drosophila, which is queen of genetics, and its experimental results by various scientists. And you re-emphasized the contributions made by few Nobel laureates, sir, and you really explained clear-cut structure of John Gurdon, who is a live example for us now. And thank you for taking the time from your busy schedule and sharing us the valuable information, even from your working place, sir. So, which shows us the dedication and which may also inspire us, sir. Thank you so much for being with us, sir. Thank you.
There is a question. Okay, so the question is how zebrafish. Yes. Can I answer that question which is in the chat box?
Yes, sir. Yes, you are.
मूल बात के बोलने से फायदा नहीं, याद नहीं रहता।
What
अब
is
जिसको
it?
पेट लिख दिए हैं, अब उसको हम लोगों के दिमाग में उतना नहीं है, वो अपने दे जाता है तो
Somebody else is.
Divakar Chatterjee is shouting.
आदमी लगा देगा, कोई उपाय नहीं।
Can you unmute him? Can you mute him, please?
Yes, sir. Unmute. Sir, you can answer the question.
Okay. So the question is how good a model system zebrafish is. So zebrafish is an excellent model system because, and also the advantage is that it's a vertebrate, and therefore many of the things that happen in zebrafish also happen in other vertebrates, including human beings. The other thing very peculiar about zebrafish is that zebrafish can regenerate its heart. So you can remove almost 60-70% of the heart of zebrafish and it can regenerate. And therefore, people are now studying how it can regenerate the heart. So you can make different kinds of injuries to zebrafish heart and it can regenerate the complete heart. In fact, in Agarkar Research Institute, we have a functional zebrafish lab, and a colleague of mine, Chinmay Patra, is looking at mechanisms of how zebrafish can regenerate heart. We know that humans cannot regenerate organs like heart and brain. If cardiomyocytes are damaged due to, let's say, cardiac infarct, then they are replaced by fibroblasts, and therefore there are chances of getting heart attacks again. So in zebrafish, people are trying to find out how it does it and whether that knowledge can be used for some kind of, relief to human patients. Good morning, Dr. Susan.
Yeah. Actually, I happened to meet John Gurdon in 1992
Fantastic.
at a symposium, and he was telling that he called Queen Elizabeth to see his tadpoles,
Yes,
the cloned
yes,
tadpoles.
Yes.
And she looked at them and she said, they are looking so dirty. And he was so disappointed because he had done so much of work and he had got it. Nice meeting.
John Gurdon is such a gentleman, a very nice person.
Yeah, yes.
Yes.
Thank you.
And he still, goes up the Himalayas. He's still so strong.
Yeah.
He likes to look at rhododendrons. He's very fond of rhododendrons.
Rhododendrons.
When he came to Pune, actually, I arranged a trip for him to Ajanta Ellora. He was very happy.
Okay.
I heard that yesterday's your talk went off very well. Rita told me, I'm sorry, I have too many things, so I could not attend that.
I will invite you for a lecture sometime.
Anytime. My pleasure. I come to Xavier's all the time.
Okay.
Okay, then I'll.
Okay, bye.
Yeah, you can start your talk now.
Thank you, sir.
Okay.
Thank you for such an informative session.
Thank you.
Now we look forward to the mesmerizing lecture of Dr. Susan Epen. Kindly take over the session, ma'am. Thank you.
Just a moment. Can you see my slides?
Yes, ma'am. We can see it.
Is it size is okay?
Yes, ma'am.
Yeah.
Madam, press F5 so that it will be full screen.
Yeah, full screen. Okay, is it okay now?
Yes, ma'am. It's okay.
Yeah, full screen. Am I audible?
Yes, madam. You are clear.
Okay, yeah. Just a moment. So today I am going to speak about plants for remediation of metals and organic pollutants and development of transgenic plants. You know that plants not only give us food and feed, but they can also protect our environment. If plants were not there, we would not have been able to live on this planet. Not only we, none of the animals would have been able to live on this planet. So we are glad that plants are there to protect us from chemicals also. Now let us go to the topic. In 21st century, there is increased industrialization, urbanization, and anthropogenic activities have enhanced the levels of pollutants in the environment. When you go outside driving in a car or traveling in a train in Mumbai or in a bus, you feel how polluted the environment is. So how can you clean up the environment? Man has polluted the environment completely. So we now need remediation methods for cleaning up of environment. Chemical and physical methods have been used so far to clean up the environment, but they are very expensive and environmentally destructive. An alternate method is use of plants for cleaning up of pollutants, and this is known as phytoremediation, use of plants for remediation of environment, and this is a green technology. And this is a very highly suitable method for a country like India, which is developing. So this is phytoremediation, use of plants for cleaning up of environment. So phytoremediation is aesthetically pleasing, economically cheaper, but the disadvantage is that it is very slow. As you know, plants have got extensive root system. Under the soil in the underground, the root system expands to several kilometers and reaches each soil, each part of the soil into crevices and all. They can move, and they are like solar driven pumps, taking up metals and other things from the soil. If you look at the pollutants, there are two types of pollutants. One is metals and second is organic pollutants. Among metals, there can be metalloids, then radionuclides, etc. And among the organic pollutants, there are man-made pollutants like xenobiotics. So is there a difference between metals and organic pollutants as far as plants are concerned? Yes. Plants can take up metals. And what will plants do with the metals? If it's a useful metal, they will use it in their metabolism to be a part of the enzyme or part of the nucleic acid or whatever. But they don't degrade the metal into smaller and smaller things. They cannot be degraded. They can be only contained. If it's a dangerous metal, for example, suppose it is cadmium, it will store it inside the plant. It is just contained in the plant and it cannot degrade the metal further. Whereas if it is an organic pollutant or if it's a xenobiotic, the plant can degrade it to some extent, not completely degrade it. To some extent, it can be degraded. And sometimes some of the things can be mineralized, and plants, once they degrade the organic pollutants, don't think that they are excreting the whole thing. They are getting stored in the vacuoles or in the cell wall. They may not get completely degraded. So as I said, there are two types of pollutants, organic pollutants and inorganic pollutants. Organic pollutants, difference between the two are, one is organic pollutants may be biodegradable or non-biodegradable, whereas inorganic pollutants like metals are not biodegradable. They can be only contained. Okay, this are also phytoremediation approach for organic pollutants is different from inorganic pollutants, because all these pollutants are immobilized, like inorganic pollutants are immobilized inside the plant cell components, whereas the organic pollutants are immobilized, whereas inorganic pollutants can be utilized by the plants. So if you look at a plant system, it has very extensive root system. And the part is hidden underground, so you don't see the root system. Then they are all branched and all that. And in the soil, there are a lot of minerals and metals and all those things. And plants need some metals for growth and differentiation, for growth and development. But there are certain dangerous metals also in the soil released by the factories and by the automobiles and other things. So there are a lot of bacteria, fungi, etc. in the soil, and these some of these fungi and bacteria help in solubilizing some of the metals and all that, and it will help in absorption by the plants. So roots are involved in absorbing the metals. For absorbing the metals, the roots have got transporters, special channels, cytoplasmic chelators, etc. So the roots take up the metals, and from the roots, they are loaded, they enter the cells. There are many cells like epidermal cells, then cortex cells, endodermis, and finally, they are loaded into the xylem. And from the xylem, the metals, they go up into the shoot biomass. And sometimes they are stored in the leaf, or they are utilized for the plant metabolism. This is called as xylem transport. Okay, and there are different types of xylem transport. Once they are in the shoot biomass, the metals will be sequestered. They can be used or it can be bound in the vacuole, they can be bound by chelation, by chelators, etc. So this is a beautiful phenomena shown by plants, the ability to take up metals and transport to the shoot biomass, and also to utilize it for the plant metabolism. So now let us go to phytoremediation of organic pollutants first. There are different organic pollutants released into the environment by anthropogenic activities or by nature itself. For example, many people, especially abroad, they use herbicides like 2,4-D to remove weeds, or some other herbicides like biolophosos or some other herbicides. So there are many pests in the plants, as I said yesterday. They are infected with many pests, and people use many chemicals like chlorpyrifos, lindane, trifluoralin, etc. And factories release many chemicals into the environment like TCE, EDB, chloroform, carbon tetrachloride, etc. And in the atmosphere, there are many polyaromatic hydrocarbons, again released by the anthropogenic activities. They include anthracene, then benzanthracene is a very carcinogenic compound, naphthalene, etc. Then there are explosives like nitrate esters like GTN, PTN, PETN, then nitroaromatic explosives like TNT, then RDX, HMX, etc. Then there are PCBs which are very dangerous. Then phenols are also quite dangerous for the plants. Then we take a lot of drugs. Not only we inject drugs into animals and for their health, and these drugs are released back into the soil and solutions, and they also cause a lot of environmental damage. So if you look at the xenobiotic metabolism in plants or an organic pollutant in plants, this take place by 3 phases. There is phase 1, this is a phase 1, phase 2, and phase 3. In phase 1, an enzyme called cytochrome P450 enzyme is very important. And in phase 2, glutathione transferase participate. And in the phase 3, membrane-bound ATP-driven pumps are taking action. Okay, now let us come to the phase 1. What happens to a xenobiotic pollutant? If it is in the soil or solution, the plant will take it up. It goes inside the plant cells. Then the molecule, because of cytochrome P450 enzyme, the xenobiotic pollutant will undergo a transformation or a reduction, and functional groups are introduced so that the compound becomes more polar, more water soluble. Then it moves into the second phase. In the second phase, glutathione transferase will act, and the product of phase 1, it will get conjugated to a sugar molecule or a peptide molecule, and the molecule will become less toxic. And glutathione transferase is acting in phase 2. And in the phase 3, the molecule which is bound to sugar or peptide, they are moved into vacuoles, or they will be deposited in plant cell wall, that is lignin or cellulose or something like that. It will become a part of the cell wall so that it is immobilized, or becomes inside, encapsulated inside the vacuoles, so that it doesn't affect the metabolism of the plant cell. But one thing I want to tell you, that as far as xenobiotic pollutants or organic pollutants are concerned, the 2 enzymes like cytochrome P450 enzyme and glutathione transferase enzymes are not very efficient, because they are having other functions also, like in secondary metabolism and oxidative stress and all that. So they are not very efficient. So we thought we will clone some efficient cytochrome P450 gene and this efficient glutathione transferase gene from other organisms and introduce into plants and see whether they will help in xenobiotic degradation further. So this is a plant cell, a root cell. Suppose this is a root cell, the organic pollutants will enter, then cytochrome P450 will act on it, and it will be then conjugated to a sugar molecule, and then it will be sequestered into the cell wall. It will become a part of the cell wall. It will get immobilized in the cell wall. It will get bound to the lignin molecule in the cell wall, so that the plant metabolism is unaffected by the xenobiotic pollutant. Otherwise, the molecule will be carried to the vacuoles and sequestered there, and it will be stored in the vacuole, and it doesn't come out, and the plant is safe as far as the xenobiotic pollutants are concerned. The similar thing will happen for inorganic pollutants, but as far as metals are concerned, there are many transporters in the cell wall or in the cell membrane, and transporters will take up the inorganic pollutants inside, and it will be moving from cell to cell. Either it will be sequestered or it will be used for plant metabolism. Now let us think about the cytochrome P450 enzyme, which is a phase 1 enzyme which helps in the phase 1 of xenobiotic degradation. So they perform oxidative, peroxidative, and reductive metabolism of both endogenous and xenobiotic substrates. So it's a super family, and especially you heard yesterday that human liver has a variety of cytochrome P450 enzyme, and human liver is very efficient in the metabolism of several drugs that we humans take every day. So this, we thought we'll clone a gene from human beings and introduce into plants and see whether they are more efficient or not. Okay, so we developed transgenic tobacco plant expressing a human cytochrome P450-2E1 gene, and we studied the degradation of some particular xenobiotic. So we, in our lab, maybe in 2006 or 7 or 8, we cloned this human cytochrome P450-2E1 gene, which is involved in phase 1 enzyme and introduced into tobacco. And there's a lindane, which is a banned pesticide. We thought we'll study whether the transgenic tobacco plant can take up lindane and degrade it further. So this is the object of the study, cloning of a human cytochrome P450-2E1 gene in plant-based binary vectors like P. cambia 1301 and P. cambia 1302, development of transgenic tobacco plants using agrobacterium-mediated transformation. Yesterday I told about the different steps in agrobacterium-mediated transformation and evaluation of plants expressing this particular gene for degradation of lindane, which is a banned pesticide. So clone the gene, then sequence was confirmed, and we are put in a plant expression plasmid with a double 35S promoter, NOS terminator, etc. And this is the vector with the hygromycin gene for selection and a GUS gene for transient expression. This is the picture, linear picture of the plasmid. So tobacco plants were regenerated, transferred to soil, and we confirmed the presence of the gene by PCR, RT-PCR, etc. for a actin gene, and this is for the cytochrome P450 gene. Then a southern blot hybridization, and we studied the kinetics of C14. We labeled lindane with C14. We used labeled C14 labeled lindane, and we studied, and these transgenic tobacco lines along with the control were grown in hydroponic media supplemented with C14 lindane, which is a banned pesticide. And we found that compared to the control, the transgenic plant could take up, they could remove more lindane from the medium within a short period of time. So transgenic plants were much more efficient. C14 lindane removal for different tobacco transgenic lines were efficient, more efficient compared to, they removed higher levels of lindane from hydroponics media compared to the control plants. So further studies, they were doing further studies and also studies on how cytochrome P450-2E1 human gene will influence, it will take up and degrade other insecticide and xenobiotics. So now we come to phase 2. I said in phase 2, glutathione transferase gene is very important, and we cloned a gene, glutathione transferase gene in the phase 2 was cloned from a fungus. Now let me come to that. So GST is a very important for a nucleophilic attack by sulfur atom of glutathione on the electrophilic group of a variety of hydrophobic xenobiotics. So earlier there is a report that Brassica gentia overexpressing the glutathione transferase showed enhanced tolerance to some of the herbicides. So and also a work in tobacco and maize that with the enhanced expression of glutathione transferase could remediate alachlor. So in our work, we found a GST gene from a biocontrol fungus, because one of us was working on trichoderma virens. Trichoderma virens can be used because it has efficient glutathione transferase. We cloned the gene from trichoderma virens, which is a fungus, and we introduced the gene into tobacco plants. And transgenic plants were tested for degradation and uptake of radio-labeled anthracene. So this is how we developed the transgenic plant, expressing a glutathione gene. The glutathione gene was cloned from a fungus Trichoderma virens, and first in plant-based binary vector Pcambia 1301, then developed transgenic plants by agrobacterium-mediated transformation, then evaluated the tobacco plants to see whether the gene is incorporated in the transgenic tobacco or not, and whether they're expressed, and if they're expressed, the plants were selected, and we tried for this benzanthrine removal from soil and solution. So this is a fungus, Trichoderma virens, from which we isolated the gene. So this is the linear representation of our gene along with others in the plasmid. This is the glutathione transferase gene with a 35 years double promoter, then NOS terminator, etc. Then there is a hygromycin for selection and a UIDA for a GUS expression, which is a transient expression assay. And we did the PCR amplification and found the amplification of TVGST gene. Then RT-PCR was done, control doesn't show. Then southern blot was done to confirm. Then we assayed the enzyme, the glutathione transferase in transgenic and controlled tobacco plants. C is controlled, and all these are transgenic plants. And G1 to G14 transgenic plants showed a higher expression of glutathione transferase, especially the G11 line. This line, G11 line, showed very high expression, almost 6-fold expression of glutathione transferase compared to the control plant. Now we wanted to use this plant for phytodegradation of anthracene. Anthracene is a molecule with 2 benzene rings. So we grew this transgenic plant. This is a transgenic plant. This is a control plant in anthracene. Labeled anthracene supplemented at a concentration of 5 millimolar, and this was grown in soil, and after a few weeks, after 23 days, the control plant did not grow, and it was showing yellowish in color, and it was not growing further, whereas the transgenic plant was very healthy even after 23 days of growth in a medium, in a soil supplemented with 5 millimolar of radio-labeled anthracene. Anthracene uptake by transgenic and controlled plants were studied, and always the removal of anthracene by the transgenic plants. This purple one is a control, or transgenic. This is a medium, like removal from medium. So control plant could not remove much of anthracene from the medium, whereas most of the transgenic plant could remove higher levels of anthracene. This is a radio-labeled anthracene, C14 labeled anthracene. They could remove higher levels of anthracene from medium compared to the transgenic plant, compared to the control plant. So then we wanted to see what happens to the anthracene once the transgenic plant take up this. So we did HPLC analysis of control plant. This is a control plant, and this is a transgenic plant. In the control plant, the plant takes up anthracene and it remains as it is. Nothing happens to the anthracene molecule after it is taken up. Now what happens to our transgenic plant? When we do HPLC analysis, you don't see the peak at the same place, whereas here you see a fresh peak. So what is this peak? So further analysis was done using mass spectrum analysis, and we found that this peak that we showed earlier, it corresponds to like a naphthalene. Naphthalene as it's anthracene, which is a 3 benzene ring molecule, is degraded into a 2 benzene ring molecule by the transgenic plant. So then in phase 3, that is about what I did in what we did in about the GST. Then phase 3 transformation is for transporting these foreign chemicals to vacuoles or to cell wall, and you need ATP binding ABC transporters, etc. So it can be worked out, but we are not worked in that area. So we can also enhance excretion of certain extracellular enzymes such as lactases, peroxidases, etc. into the soil. We have done some work on phenol remediation using plants, and we found that the plants excrete peroxidase into the medium or into the solution or into the soil, and they can polymerize phenol. So there are some reports from other people that peroxidase, introduce a transgenic peroxidase, could remove phenol from the medium at a higher efficiency. So there are so many reports like that. Now let us move on. This is what I am talking about, degradation of organic pollutants or xenobiotic pollutants. Now let us move to metals and radionuclides. Metals, metalloids, and radionuclides, plants need mineral nutrients for their growth and development. 13 mineral elements like molybdenum, copper, zinc, manganese, iron, boron, chlorine, sulfur, phosphorus, magnesium, calcium. Some of them are micronutrients, some are macronutrients, nitrogen, etc. needed, sometimes silicon and sodium. And the uptake of mineral elements from soil by the plants is a very selective process. But some minerals may be discriminated by the transporters. So roots are directly in contact with soil, and the soil contains the mineral elements, and the plant can take up the mineral elements from the soil. So how does the mineral elements move into the soil? There are two types of process. One is a passive process, and other one is an active process. In the passive process, the primary cell wall, you know that primary cell wall consists of network of cellulose, hemicellulose, and glycoprotein, and contain pores like that, 35 to 38 angstrom. And plasma membrane have got a selective capacity to selectively take up metals. This is an active transport because ATP is involved. Okay. So types of metal transport. In one type of transport, it is an apoplastic type of transport. What is apoplastic type of transport? You know that yesterday I said that plant cells contain plant cell wall. So once it enters the cell wall, it need not enter the plant cell inside by crossing the plasma, crossing the plasma membrane. It can move from cell wall to cell wall. So cell wall to cell wall movement is called apoplastic type of movement. And what happens when metal moves from cell wall to cell wall in the root? It will go from epidermis, if you remember the structure of root, it will go to epidermal cell wall, then it will go to root cortex cell wall. But when it comes to endodermis, what is endodermis? Is a very thickened type of cell. It is suberonized cell. When it comes to endodermis, all the metals are stopped. They do not get into the xylem, and they are unable to move to the top of the plant, to the shoot biomass. This is the apoplastic type of movement of metals, cell wall to cell wall movement, and it is stopped at the root endodermis because the root endodermis has got very thickened cell walls. So they do not, because of this movement, the metals do not get loaded into the xylem and do not move into the shoot apex, shoot and leaf biomass. In the second type of metal transport, which is symplast, cell-to-cell movement through the plasmodesmata. There is a, you know about plasmodesmata connect cells, so it goes inside by active transport, and it crosses the endodermis also. Is that a thickened root is not a barrier for a symplast type of movement. It gets loaded into xylem and moves by translocation, goes to the shoot and the leaf, and in the leaf cells or the shoot cells, they get sequestered or they will be utilized for various metabolism. So now, toxic heavy metals by roots, transport of toxic heavy metals, because some of the heavy metals resemble they have same ionic properties of essential and non-essential metals are sometimes very similar. Chemically, they look very similar for the plant. So the plant transporters which actively take up these metals cannot distinguish between two metals. So by mistake, they take up toxic heavy metals. So transporters having broad selectivity, specificity for essential and non-essential elements cannot at times distinguish between an essential metal and a non-essential metal. For example, a zinc transporter cannot distinguish between zinc and cadmium. So when cadmium is present in the soil, the transporter which is supposed to transport zinc can transport cadmium, which is supposed to be a very toxic metal. Now, let us go and see what is hyperaccumulators. Hyperaccumulators are a small group of plants which accumulate metals at a very high concentration, that is 0.1 to 0.0% of dry weight in the shoot biomass. It is not in the root biomass. Root biomass, any plant can maybe take up by a apoplastic type of movement, but hyperaccumulators will accumulate high concentration of metals like nickel, cadmium, etc. in the shoot biomass. But unfortunately, these plants are very low biomass plants. They have such tiny plants and they occur only in maybe sometimes in Europe or somewhere, and they live only for 1 or 2 months. So you cannot use it for actually for removing the metals from the soil. You know, they are very small in biomass. They are limited to some areas in the world only, and plants are very small, and they are limited in their growth period also, just maybe a few weeks or something like that. So we have to have a candidate plants which should accumulate high levels of metals in the shoot biomass. So phytoremediation can be used for improving its uptake of metals by using transgenic technology also. So now we selected 2 genes from the bread mold, that is a fungus, Neurospora crassa. We took up zinc transporter gene and a copper transporter gene. You know, zinc and copper, they are like 2 sides of the, I mean, zinc is like, you know, it's a useful metal, as well as at a high concentration, it can be toxic also. Copper also, it's needed for the plant metabolism, but high concentration, it will be toxic. So it is like a 2 sides of a coin. So we have developed transgenic plant with a zinc transporter gene TSWREIS1 from Neurospora crassa. We cloned this gene from Neurospora crassa, a bread mold, and introduced in the tobacco plant. In another experiment, we cloned a gene from Neurospora crassa, that's a copper transporter gene, copper T, and introduced into tobacco. So the transgenic tobacco plants expressing a zinc transporter gene showed enhanced zinc acquisition. So we cloned the gene from Neurospora crassa, and in plant-based binary vectors like PCAMBIA 1301, then we developed transgenic plants, tobacco, Nicotiana tabacum, expressing a TSWREIS1 using Agrobacterium tumefaciens mediated plant transmission method, and we evaluated the transgenic tobacco expressing this Neurospora crassa TSWREIS1 for uptake of zinc to study how the plant will behave for zinc uptake, so that we can use either for phytofortification or phytoremediation. You know, phytofortification is and phytoremediation are like 2 sides of the coin. You need zinc for at least 300 enzymes in our system, and it can be used for phytofortification. And when the concentration is very high or something like that, you can use for phytoremediation. So this is the zinc transporter gene we cloned from Neurospora crassa, say TSWREIS1 NCU07 62.3. This is cloned from there. This is the linear representation of zinc transporter TSWREIS1 with a 35 years double promoter and a NOS terminator. So linear representation of tDNA of a tDNA containing this particular gene, okay, with a 35S double promoter and hygromycin as a selectable marker, and Gus gene or UIDA gene for transient expression. So when we do histochemical analysis, you see spots just in 48 hours after the experiment. You can see blue spots. You can even later you can see after many days also you can see the transient expression, which shows as blue color. You can even use GFP gene, which will give a fluorescent color, fluorescent nature under a fluorescent microscope. So you can use Gus gene or GFP gene or whichever gene you want to use, you can use. But in our case, we use this UIDA gene, which gives a blue color on staining. Then the zinc transporter, as usual, we characterize by PCR confirmation, then by RT-PCR, etc. Then southern blot was also conducted to find see that this is stably integrated into the plant. Now we wanted to use this particular transgenic tobacco plants expressing zinc transporter from Neurospora crassa for uptake of zinc. So several experiments were conducted. These are the plant lines grown in the presence of zinc. And zinc accumulation by roots. This is the accumulation of zinc by roots, and this is accumulation in the shoots. So all the transgenic lines which are shown in purple color, they accumulate more zinc in the root compared to the control, which is shown in blue color. Similarly, all the shoot structures of transgenic lines showed higher accumulation of zinc compared to the control. We studied the zinc accumulation by the transgenic plant at different concentration, and from 0.7 to 700 micromolar, and at all concentrations the transgenic plant showed better uptake of zinc in both roots as well as in shoots. So transgenic plant could take up more zinc. They could not only take more zinc from the soil and solutions, they could also transport it to the shoot. That is quite important. So then we wanted to see whether these plants can take up cadmium. I told you in the beginning that these transporters sometimes cannot distinguish between cadmium and zinc. So we found that when cadmium was also supplied, there was our transgenic plant, they did not take up more cadmium compared to the control, both in root and shoot. That means this particular zinc transporter is more specific to zinc rather than cadmium. It's a good transporter for taking up zinc because it did not take up the dangerous toxic metal cadmium. So we did some ECEM EDAX studies. This is the images of roots of transgenic plant treated with the 700 micromolar of zinc. This is the root tip and this is the middle of the root. And then from this scanning, this analysis, we looked how much of zinc is present, in these cells. So we found that, in using the EDAX studies, we found that transgenic plants had a higher zinc in these cells at high concentration compared to the control. I cannot see here. Okay, at high concentration of zinc, transgenic plant showed higher level of zinc in these cells compared to the non-transgenic plant, which did not show any zinc. So what is the conclusion? We transferred both the zinc transporter gene and a copper transporter gene from Neurospora crassa into tobacco. So what is the result? Well, conclusion is that the transgenic tobacco plants with the fungal zinc transporter gene could take up almost 11 times more zinc compared to controlled plants, and this could be transported to the shoot masses also. And most of the zinc was located in the roots, and a good amount was transferred to shoots also. So we also did development of transgenic plants of fungal copper transporter gene for enhanced uptake of copper. Again, the gene was cloned from Neurospora crassa, and this gene details are given here. This gene is called as TCU1. So using first we transferred it to a plant binary vector. P can be a 1301 and P can be a 1302. Then we developed the transgenic plant using agrobacterium mediated transformation method, and we evaluated the transgenic tobacco plants for uptake of copper from solutions. So again characterize them by PCR amplification, RT-PCR done, southern blot was done, then uptake was studied, and most of the transgenic plant to take up more copper from a medium compared to the control. This is a control, but the most of the transgenic plants, and especially some lines, could take up more copper from the medium at different concentrations. So this is another concentration. This is a higher concentration. Transgenic plants mostly did better, especially some lines like this particular line, C24 did very good compared to even other transgenic plant. So, transgenic tobacco plants expressing a fungal copper transporter gene from Neurospora crassa in general could remove more copper from solution more efficiently than control plants. So, and most of the copper taken up by plants remained in roots, but only very little was translocated to the shoots. And we studied the Mendelian inheritance of this character. I spoke about Mendelian inheritance yesterday. They are segregated in 3:1 ratio, and it is transmitted to the progeny. Okay. So copper transporter is a very good promising candidate for developing transgenic plants for enhanced removal of copper from contaminated bodies. So the present studies have shown that transgenic plants with a TSWREIS1 gene could take a more zinc compared to control. Similarly, transgenic plant with a copper transporter gene could take up more copper from soil and solutions. So both zinc and copper transporter, copper are important micronutrients. So you can use it for biofortification of food crops as well, not only for phytoremediation, but for a biofortification also. Now we developed some transgenic plants of tobacco using a nickel cobalt transporter gene. The objective was we had a lot of cobalt 60 waste in a bioatomic research center where I was working, and we wanted to see whether plants with a nickel cobalt transporter gene could take up more cobalt 60 from a cobalt 60 waste. So again, we cloned a nickel cobalt transporter gene from a bacteria this time, R. polystris, and agrobacterium mediated transformation was done. Plants were regenerated and characterized as usual, like with a PCR and RT-PCR and southern blot and all those things. Then we studied the nickel cobalt transporter, whether this plant expressing this nickel cobalt, bacterial nickel cobalt transporter could take up more cobalt 60 or not. So this is a characterization of the nickel cobalt transporter gene using a PCR analysis. Then we studied tested for uptake of cobalt 60, and we found that transgenic plants showed higher uptake of cobalt 60 compared to control plants. Most of the cobalt was accumulated in roots, and only 0.3 to 3% was translocated to shoots. So this is the control plant, and this is a transgenic plant, and transgenic plant to take up more cobalt 60, and they were grown in cobalt 60 waste, and we could find that the transgenic plant expressing a nickel cobalt transporter gene from a bacteria R. polystris could take up more cobalt 60 compared to control non-transgenic plant. So this is a non-transgenic plant. After growing in cobalt 60, the control plants do not take up cobalt 60 and they remain green, whereas the transformed plants take up cobalt 60 from the waste and they remain little yellowish and unhealthy in color. So we also studied other radionuclides like strontium 90. then cesium-137, plutonium, americium, uranium, etc. And they were used for remediation of many of these radionuclides from solutions, and also from low-level nucleoactive radioactive waste like Calotropis gigantea, vetiver species, vetiver cyanoids, Chromalina odorata, Catharanthus roseus, etc. So all the plants could take up strontium 90, cesium, this should be cesium, cesium-137, plutonium, and americium. So now let me go to vetiver grass, which is a miracle grass, Vetiveria cyanoids. It's known as Vetiveria cyanoids. You may be knowing this plant. It is grown in Karnataka initially by the farmers, traditionally grown for environmental conservation to prevent soil erosion, waste conservation, and wastewater treatment. And this plant is also used for flood control, and it is a source of aromatic oil. You know, plant is called Kas in Hindi, and in Malayalam it is called Ramacham. This plant can tolerate high alkalinity and acidity. And one advantage for this plant is, it is vegetatively propagated. It doesn't have seeds, especially the varieties which are grown in South India. The roots are really very, very long, and they reach 6 to 7 meters down the earth. So we developed a tissue culture protocol so that we could get large number of uniform plants, like 10,000, 20,000 plants in a short time in the test tube. And these plants were very uniform in size. They were manageable in the test tube. So we could study the uptake of radionuclides, metals. We could use them for studying degradation of organic pollutants. So we use some phytoremediation experiments. We did some experiments. How do they survive in our uranium mill tailings under laboratory conditions? How they survive under various effluents from UCIL and other tailing ponds of uranium. After uranium extraction in Jedguda, we have large tailing ponds 1, 2, and 3, and we screw them there and studied how do they survive in such ponds and whether they take up uranium or not. Their morphological studies, physiological and ecological characteristics with relevance to soil conservation was studied. This is a depletion of recovery of uranium at low concentration. First, we studied low concentration, and the plants could take up in a short time, after 2 days or 3 days, the plants could take up almost 90 to 100% of uranium at low concentration. So what was the observation? When we grow plants at a low concentration, the plants at low concentration, they take up uranium and they survive very well. But even at high concentration, like 238 ppm, plants do not die. Older leaves may die, but younger leaves will come and they still go on taking up uranium. Even after 50 days at a very high concentration, that is 500 ppm of uranium, the plants can still survive. And even at 1000 ppm or more, plants can grow for some time and they will die within 15 days. So we tried different concentration, 1000, these are the low concentration, 318, 619, 927 ppm, 1263, and the plants do take up uranium as the days progress. But at high concentration also, even when the plants are dying, still they deplete uranium from the medium. This is a medium containing uranium, and this graph goes up even at high concentration of 11,900 ppm of uranium. Plants still take up, even when they are dying, they are taking up uranium. So this is a uranium recovery. We could take out the small plants growing in uranium and we could extract back the uranium. For example, at 318, when the plants are exposed to 318 ppm of uranium, we could get 75%, recover 75% of the uranium from solution using the plant, so that at higher concentration, at a very high concentration also, we could get almost 50% of uranium. We could extract the uranium from the plant. You can see the picture here, root and shoot, like uranium concentration, how much is present in the root. Root is shown in red color and shoot is shown in green color. and the concentration of uranium, when as the concentration goes up, the shoot biomass take up more uranium compared to the root biomass. So this is sort of a autoradiographic study using plants which were exposed to uranium. This is the control plant. In the middle, you can see the control plant. This is autoradiograph of the same one. So here you don't see any image. That means there's no uranium. Whereas when you use a low concentration of uranium, most of the uranium is in the root or in the lower part, as you see from the image. Whereas when you use high concentration, most of the uranium not only in the root, but it is translocated to the shoot. This is a phosphor image analysis. Then we recovered uranium from the plants, small plants after acid digestion. You can see the yellow metal here, here, and here. Then I said that we have a Department of Atomic Energy after it isolates uranium, extracts uranium, low concentration of uranium, which cannot be extracted further. A very large amount of uranium after extraction, very large amount of waste after extraction of uranium, and this contains very low concentration uranium, are dumped in some 3 mill tailing ponds in Jedguda, and we brought the mill tailings to Mumbai, and we grew the plants in different mill tailings, and we wanted to see whether our plants could survive in that. And 82 days after transplantation in Jedguda mill tailings, the plants were happy and they were surviving. Even after 1.5 years of transplantation, and we don't even provide them water, still they can survive. This is the control plant, and mill tailing soil in the laboratory, they could survive. So now we also studied phytoremediation of cesium, strontium, etc. And this is the cesium. This is cesium, and this is the strontium. The strontium is taken up by vetiver cyanoids. Cesium is also taken up as days go by. The plants take up time in hours. In 168 hours, lot of strontium is taken up. 90% of the strontium is taken up, and maybe 50% of cesium is taken up by the vetiver cyanoid plants. So then we studied the distribution of strontium and cesium in shoot and in root. As far as cesium is concerned, most of it is in the root and with a little translocation to the shoot, whereas as far as strontium is concerned, most of it is translocated to the shoot, and in the root there is a comparatively less amount. So this is time in hours. We grew this plant in low-level nuclear waste. These plants were grown in low-level nuclear waste, and the activity decreases from the medium after 360 hours. It almost comes to 0. That means the plants are taking up the radioactivity. So the physicochemical analysis of low-level nuclear waste. Then we wanted to see how this plant will survive different waste from different mill tailing ponds and other after uranium extraction, TRICS waste, etc. from BRC and Uranium Corporation of India, etc. And we brought these different, solutions, effluents from different places, like ETP effluent from BRC, barren effluent before treatment, neutral tailing slurry, Batinda mine discharge, then ETP outflow, Jedguda discharge, etc. In most of the things, the plants could survive very well for quite a few days. Only in this particular one, barren effluent before treatment, the plants were dying. So we tried several strengths in the barren effluent where the plants were dying. We diluted the solution. The plants, 80% they could survive. 20% they could survive very well. Control plants are here. So only at 100% there is a plant death. So we studied the different radionuclides radionuclide uptake by vetiver plants in TRICS waste. This is the waste from particular plant, and this is antimony, ruthenium, and cesium. And both root and shoot and total amount of radionuclides are shown here. So antimony, radioactive ruthenium, cesium, all could be taken up by both vetiver cyanoids, and it was translocated to the shoot, and some of it remain in the root. Okay, and this is a total. Blue one is a total one. Now we wanted to see how the plants will survive in the vetiver. Earlier I said the milk tailing after extraction. Now how will it survive in the uranium ore which contain uranium? So we brought the ore from Jaduguda, and like a plant could grow in the ore, and when you add citric acid, plant could extract more uranium from soil and translocate into the shoot. So this is a ruthenium nitrate after growth for a few days, 20 days. Then this is a UAD effluent. Then we also did some anatomical and ultrastructural studies of vetiver plants exposed to uranium. What happened to the leaf? This is exposed to very high concentration of uranium, like 1000 ppm of uranium. And this is a control leaf. And this is a uranium exposed leaf tip, middle part of the leaf. There are some structural changes, but not very drastic change could be seen in plants exposed to 1000 ppm of uranium. This is the lower part of the leaf, and here is a plant with exposed to uranium. Similarly, we also saw the root. In root, there were certain sort of distortions when 1000 ppm plants were exposed to 1000 ppm. You can see here, now this is a control one, and this is a plant with exposed to uranium, 1000 ppm uranium. There is some sort of distortion in vetiver roots exposed to uranium. Also, we did some EM or electron microscopic studies. Now, I also would like to tell a little bit about using this plant for a sustainable environment management solution using bioengineering techniques. This I retired in the year 2011, and at that time we had planted these plants in our Jaduguda tailing ponds. And I don't know what's the result of that, but I will tell. I have borrowed a few slides from one of my friends, Dr. Pradeep Kumar from Calcutta. He is using these vetiver plants for sustainable environment management solutions. So he wanted to restore, Dr. Pradeep Kumar wanted to restore some canals and other and other lakes which are contaminated with pollutants. So he grew these vetiver plants as a floating garden, as a floating in floats in these lakes which are contaminated. This is a project in West Bengal for restoration of canals, water bodies, and lakes. So this is canal eco restoration. Canal eco restoration in Calcutta. Here, near the banks of the river, Mr. Pradeep uses biological material like coir, like then jute, etc., to protect the banks of the river. And then he grows canna and other plants also, along with the vetiver plants in the lakes. So this work is done in collaboration with Irrigation and Waterways Department of Government of West Bengal for restoration of canals, water bodies, lakes, and river embankment stabilization. When there is a flooding, you want to stabilize the land on the banks of the river and the lakes. So this is also a water body eco restoration. This looks very dirty, and this is used for restoration. This is also in West Bengal, Jaya Shri Textiles Trishra, this project from Birla Grasim. So in water body restoration, he grows these plants, vetiver plants, as floats in the area, in the lake, which is supposed to be contaminated. Then after some time, he will take up these plants, and he will incinerate the plant, and fresh plants are put in floats. This is a floating garden or a biophytoponic solar aeration. Aeration is also given. This is connected to a solar system and a floating garden system with solar panels, battery bank, aerators, LED lights, etc. and automated control systems. And these plants are, vetiver plants are put as floats in these lakes, and after a certain period they are taken out and they are incinerated in incinerators and the ash disposed of, and fresh plants are put. That is how remediation is done. So this is a lake where we had gone 2 years back. This lake was supposed to be very dirty in Calcutta, Patoli Lake rejuvenation in Calcutta. And this was done by consultation with KMDA, Calcutta Metropolitan Development Authority. So the lake was very dirty, but after cleaning up with this technique, the lake became very beautiful, very clean, and on the banks of the lake, so many shops started coming, and people started coming, visiting these stalls to buy their vegetables and fruits and all that. This turned into a beautiful tourist spot after cleaning up, because many of our lakes and rivers are very dirty. This is the same lake, in Calcutta. After cleaning up, see how beautiful they are looking. It's all lighted up, and vetiver plants are being grown here. But people come here to do shopping and to buy their daily needs. Lake is rejuvenated by floating islands. In the floating island, vetiver plant is kept, and bioengineering solutions are also incorporated to make it more beautiful. Fountains are there and lights and all are there. So in Mumbai, we have a lake called the Methi River, which is supposed to be very polluted, and it needs cleaning up. So Mumbai Metropolitan Region Development Authority, MMRDA, has started a collaboration with the Pradeep's lab at it's known as BioSafra Foundation. It has a collaboration with the Netherlands also. And this Methi River is very dirty. And now they are planning to clean it up with the vetiver plants also, with some other bacteria and other things. So they are doing the initial work. River rejuvenation of floating islands and bioengineering solutions of Methi River in Mumbai. This has started, but because of the COVID, it is getting delayed. So here is Mr. Pradeep standing on his reclaimed river banks. He puts his coir and bamboo and bamboo rods and everything here, and makes us land quite stable, and puts vetiver plant for stabilization of the soil. This project is a irrigation and waterways department of a government of West Bengal, and the project site is Bankura, West Bengal. 20,000 square meters are being planted with these vetiver plants, and also with this material for protecting the from flooding and other things. So the vetiver plant stay as a bio curtain, like protecting the area. So what should we look for a candidate plants for metal acquisition? Our plants should have, when we use a plant for a metal acquisition, the plant should have the ability to take up metals to the shoots. Accumulating in the roots is not very important, because you can't pluck it off. When you are cutting away the shoots, fresh shoots should come up. Preferably it should be a grass type of plant, because like a grass have got tillering ability, and you get multiple shoots, like vetiver, many shoots will come up from the sides, even if you cut off the main shoot. And when the pollutant go to the main shoot, you can remove the shoot, and fresh shoots will come up. The plant should have the ability to accumulate metals in the shoot biomass, not only in the root biomass, because removing the root will be at times difficult if it's a soil. So, and then the plant should grow fast, and not like the hyperaccumulator, which I said, the plants have got very short biomass and they grow very slowly and they survive only for 1 month. They should have high biomass, then they should have fast growth. Then they should have tolerance to different metal concentrations. Once they take up the metal, the plant should not just die, it should have tolerance to the metals which are taken up. They should have wide spread root systems. They should grow in a required environment. Suppose you are having high temperature, the plant should be able to grow in high temperature. Suppose the medium is or the solution is a solution or river bank is alkaline or acidic in nature, the plant should be able to grow there, and it should be easy to harvest plants. And we should be able to cut off the old shoots and fresh shoots will come off. And once you cut off the fresh shoots, which have got a lot of contaminants, you should take it out separately, and you should incinerate the material in special incinerator, and you should dispose it in boxes or coffins or whatever you want to dispose it in. Then it should not be crop plants. When you select a plant, candidate plant for metal acquisition, it should not be a crop plant. It should not be consumed by humans, animals, birds, and insects. It's very important that the plants that you use for phytoremediation should not be eaten up by humans. It should not be crop plants. Even animals and like a goat and cow and other animals should not be eaten up. And birds and insects also should not stay on that and eat up the material in which are used, the plants for which we are using for phytoremediation experiments. So what are the conclusions and thoughts? This is the last slide, I suppose. We should understand better, at the moment, our knowledge on metal uptake, transport, and sequestration by plants is not very well known. We don't know much about the genes. Only when you know about the genes, we can transfer the genes. We should have very good knowledge about the different genes which are involved in metal uptake in the root, in the metal translocation, metal sequestration, etc. We should have a good understanding of the biochemistry and genomics and everything of metal uptake. Then organic pollutants, biotics. There are several pathways. All these pathways have to be worked out. At the moment, we hardly know about anything. Maybe we know about bacterial xenobiotic degradation, bacterial or fungal degradation of organic pollutants, but we hardly know anything about plants. So serious studies required to find out which are the endogenous genes involved in detoxification pathways. Then only we can improve our plants. Then we have to select endogenous plant species with a high remediation potential. We cannot bring plants like Arabidopsis halleri, which accumulate high amount of nickel from Europe, because the plant is not going to survive here. We have our own species with high remediation potential, and I think there are very few studies on remediation potential of our plants. Then once at the moment we know a little bit about the bacterial genes and fungal genes and other things, but if we know, we can introduce genes in our endogenous species, and then we can grow the plants by the river banks or by inside the lake as floating gardens or anything like that. And what are the enzymes we want to alter by mutagenesis to give better activity towards xenobiotes? How can you have a better degradation of xenobiotics? Then there are so many microorganisms, mammals, etc., which have got efficient genes for xenobiotic degradation, which plant may not be having, and we can transfer these genes from these organisms here. Now we can even go for a genome editing. As I said earlier, this slide was prepared in maybe 2011. So that time genome editing was not found. Maybe I have not added anything because hardly any work is also done in this field, in this area, using genome editing. Genome editing came into existence only in 2013. So you can even using genome editing, maybe some of the cluster of genes involved in the pathway can be added. But first we should know what the plant is doing. We should know the biochemistry of the plant, the pathway in the plant, how these genes are involved in taking of metals and transporting or degrading pollutants. Then only we can improve. So if you are using transgenic plant, you should not have antibiotics in that. Then if you are doing transgenic plants, maybe chloroplast transformation to prevent pollen transfer of transgenics. The vetiver plant that we are having in South India, this is vegetatively propagated, so it will not have pollen transfer, so that it will not go into other plants and things like that. I think that's the last slide. Thank you for your attention. Thank you all.
Thank you so much, ma'am. Now I request the participants, those who have queries, to express your queries, and the question is open for, sorry, the session is open for queries now.
We can use even genome editing to improve these plants, but first we should know the genes involved. Then only we can manipulate, edit the genes. So a lot of information is required in our candidate plant, like vetiver cyanoids. Then only we can manipulate or we can improve. Otherwise, it will be like a blind man looking at an elephant and imagining things.
Madam, there is a question in the chat box that, as you have mentioned, plants that uptake these pollutants should be disposed safely. How would they be disposed, ma'am?
They will be taken up and they'll be taken out. They'll be dried in oven or whatever way, and then they'll be incinerated. You know about incinerators in hospitals and all that, like that incinerators. They'll be incinerated so that the volume will be reduced. And if it's a very dangerous metal, we can dispose it metal in a box or something like it underground or something, or store it somewhere. And sometimes the metal will be very important, like suppose the plant is taking up gold, we can extract the gold from the plant. Okay.
Okay, ma'am. Thank you. Still any questions to be asked? Okay. Thank you so much, ma'am. Since 2 days, you are sharing the valuable information by mentioning journey of biotechnology, its advantages, developments, transgenic plants, genetically modified plants, smart agriculture. Even you have stressed on the future agriculture and safety measures like, and today even you have stressed on phytoremediation, madam, which is a very interesting topic actually. And you even stressed on the efficiency of transgenic plants, which absorb heavy metals as well as radioactive metals like cobalt 60 by quoting examples like vetiver grass, which is called a biocurtain to help eco restoration in various polluted lakes and rivers in Calcutta and Bombay. You have cited very good examples and live pictures as well as, ma'am. Your session was quite interesting, madam. It's a pleasure to have you with us, ma'am. Thank you so much.
I just would like to tell, that the Fukushima accident took place in Japan,
Yes, ma'am.
2009 or something like that. Actually, they contacted me. They contacted me to suggest some solution for removing low radioactive waste from solutions, contamination from solutions.
Okay, thank you, madam. Actually, your work is quite interesting, madam. Through your resume, we got inspired and we were awaiting your lecture, ma'am. Thank you so
Thank you.
much. And your energy levels since yesterday were really awesome, ma'am. Thank you, ma'am. Thank you for being with us.
Thank you.
ఇది చేతిని బట్టి వాయిస్ వస్తుంది అని అర్థం చేస్తున్నాను ఇక్కడ ఉంది
What time is it starting? Again, 1:45.
Ma'am, now let's all break for lunch and assemble back by 1:15. The next session would start by 1:30.
1:30. Okay.
Okay, ma'am. I request all the participants to break for lunch and assemble back by 1:15. I request all the participants to break for lunch. Yes, and you can log in again back with the same link. Thank you.